All right, I think we're going to go ahead and get started. So just a quick word before I introduce today's speaker. This is actually the last NanoEdTech seminar for this semester. We'll meet again on starting in January, on January 23rd, and check your email for announcements of the upcoming schedule. So it's a real pleasure to have as our final speaker this semester, Wilbur Lam. His resume is extremely long. I'm going to give you the brief version. He got his bachelor's degree at Rice University and then his MD at Baylor. He did a residency in pediatrics at UC San Francisco and then a PhD at UC San Francisco and UC Berkeley in bioengineering, and then a postdoc at UC Berkeley. He is currently the Paul Bowers Research Chair and Professor of Pediatrics and Biomedical Engineering at Emory and Georgia Tech. He's also Associate Dean for Innovation in the Emory School of Medicine. He's also a clinical pediatric hematologist oncologist at Children's Healthcare of Atlanta. And those are just kind of the official titles. You can see them, some of them on there. The unofficial ones are, well, they're official, but he is the PI of the Atlanta Center for Microsystems Engineers Point of Care Technologies, which is an NIH center. He's also director for the Emory Center for the Advancement of Diagnostics for a Just Society, the Just Center. He has won a number of awards, some of them very recently, as in like announced today. As of today, he is a member, well, maybe not today, but as of announced by Georgia Tech today, a member of the National Academy of Inventors, and earlier this year was announced as a elected member of the National Academy of Medicine. He's also a member of the American Society for Clinical Investigation. He's a fellow of the American Institute for Medical and Biological Engineering, and on and on and on. And with that, I will turn it over to Wilbur to talk to us today.
Great. Thank you, David. Can you guys hear me okay? You guys, okay. So thank you for having me. It's always great to talk to my family in the IEN. And so what I'll do is give you a high level, well, let me take a step back. You know, when I remember when I was a trainee, quick show of hands. Who's a trainee in here? Still getting students, PhD? Undergrad PhD student or PhD student? All right. So back when I was a PhD student, I remember this really wise mentor of mine, Nobel laureate, said, you know, to give a really good talk, what you should do is talk about just one thing and really dive into it rather than talk about a whole bunch of things and only go superficially. I'm going to do exactly the opposite of what he just said. I'm going to go do a bunch of things, do really superficially, and hopefully you guys can catch up. Feel free to stop me anytime and ask any questions. We're a small enough group here. But really what I wanted to do is give you a sense of what we do in the lab. And there's quite a bit of things, and any one of these projects I could spend an hour talking about. All right, let me see if I can advance here. All right, there we go. Okay, so what do we do in the lab? We are very interested in blood. That is what I am as a clinician. I see patients with blood disorders and cancer, but we're also an engineering lab. This is why we're here in the IEN. So we spend a lot of time making microfluidic devices. We do a lot of stuff in the microvascular and chip side. We're even starting to develop our own analytical software. Very interested in translating our technologies into new diagnostic tests and even therapeutic platforms, which I'll talk about at the end, and then bringing it back to patients that I see. So this kind of never-ending loop is the space in which our lab lives. And you know, if I really take a step back and think about how I even got into this, I'll talk about this part of blood right now. So there's several parts. There's three main aspects of blood. There's white blood cells, there's platelets and blood clotting, and then there's red blood cells. I first started my career really looking at white blood cells and started, I remember when I was a brand new grad student, I was still a medical trainee at the time, and I saw a particular patient. Now this is what's called a blood smear. And this is a normal blood smear. So what you see here are a bunch of red cells, and those little blue things are actually white blood cells. So this is a typical concentration of a white blood cell count. And a patient came in. I remember being on call, and I was still trying to figure out what my PhD project was going to be. A patient came in with a blood smear that looks like this. Now, I presume none of you are physicians, but you see that it looks really different than what I just showed before, right? You look at the white cell count, 4 orders of magnitude higher than typical, right? This is not normal. This was unfortunately a 3-year-old child. I still remember the child's name. He was diagnosed with acute lymphoblastic leukemia and unfortunately died and passed away. So he died from this process, something called leukostasis, which is kind of a stroke-like disease in which there's microvascular occlusion of leukemia in the really, really small blood cells. And it happens in the brain. You see a brain scan here, and typically it happens in the lungs as well. So I was obviously devastated that this patient died, but I was also thinking, wow, what do we know about this stuff? What does the literature show? Not much. In fact, when I traced the literature, I said, okay, it all comes back to this one paper published in the 70s. And what this guy did was he took a bunch of different leukemia cells, and he took a pipette, and he just sucked on them. He literally just sucked on them. He did it maybe 20 times on 20 patients. He actually published a paper even before this about sucking on cells. He published it in Nature in like the early, even before this one. So it was a lot easier to publish in Nature back then, apparently. So, but that was it. That was the state of the art. So what I have here is an entire PhD on one slide. So there's hope for you, right? It makes me sad, but to have everything in one bar graph. But what I did during my PhD was really explore this question. I said, okay, we know that cells from different leukemia patients, when they get sucked on a micropipette, there's different results. Why don't I quantify the actual stiffness of leukemia cells? And that's what I did. I had a small cohort of patients, so these were all kids with the same type of leukemia, all called pre-B cell acute lymphoblastic leukemia, and a handful of patients that you see on the left here. These patients had symptoms, these patients did not. Same type of leukemia. The patients with symptoms had a much higher elastic modulus, or at least an apparent elastic modulus, because it's not easy to get a true elastic modulus on the cell. So that was intriguing, and intriguing for us clinically. Like, this could end up being almost a biophysical biomarker to predict which patients with leukemia get this complication, which don't. It's actually a pretty rare complication. It only happens in about like 1 to 5% of patients with leukemia. But when it happens, it can be devastating. So published about 4 papers as my PhD, all kind of based on this. So atomic force microscopy, if anyone's ever used it, I presume some of you guys here, very sensitive technique, but also a huge pain. Not very fast either. So one question I had as a grad student was using microfluidics. Could we actually use microfluidics to study blood diseases? This was the very first microfluidic experiment I ever did, and I made it to look like small blood vessels on the size of small blood vessels. Each of those little things there is about 5 microns. And then I took white blood cells, which are also good leukocytes, from a healthy subject, i.e. my grad student friend sitting next to me. So in grad school, no one wanted to sit next to me because I would always poke them for blood. So what it looked like was these little cells squeezing through. And I was pretty fascinated by that because that's actually what happens in the body, in vivo. I said, okay, we got something here. So we published a couple of papers when I was a grad student, really kind of characterizing microfluidics and use of blood cells in microfluidics. And I'm very happy, if I can fast forward now, that I have my own trainees who've kind of picked up the baton and kind of started to explore this question using microfluidics themselves. So Kendall Williams, a grad student, actually who just got her PhD, so she'll be walking this weekend, right? That's when graduation is, right? And Dr. Music, who is a pediatric hematology oncology fellow, who now works in Alabama. So they are now using microfluidics to ask some of these similar questions. And it's much higher throughput. And what you see here on the right are leukemia cells getting jammed up in these devices. And what Kendall's done that's really smart is she's designed this device so that same blood sample can be run at different flow rates, different shear rates in these different channel sizes and shapes. So more to come on that. Okay. So now I'm going to change gears a little bit and talk again about how microfluidics can be used in the setting of cancer and specifically leukemia. So there is this concept in clinical oncology called precision cancer medicine, precision oncology, or also called personalized medicine, where the premise is this. It's been around for a few decades now. Like old school way to treat people is you got one disease diagnosis, and every patient gets the same stuff, right? One size fits all approach. We know that everyone with cancer is a little bit different, and what the desire now is the current approach is for personalized therapies, right? We take biomarkers and we take the tissue samples and we look at what's specific about this one patient's cancer, and we use biomarkers to determine, okay, we'll change it up a little bit. Maybe everybody gets slightly different medications, a slightly different regimen. What we really want is not only biomarkers, which are kind of surrogate markers of the disease, but really look at how the disease cells fundamentally respond to drugs. In the case of cancer, it's chemotherapy, and it's a response to chemotherapy, which is do these patients' cells die in the presence of the chemotherapy that you want to give the patient? So a clinical need exists for these functional screening assays, ideally that are high throughput, that you could measure a bunch of different conditions, but also rapid enough that you could use it clinically. I mean, it doesn't have to be like immediate, what's called point of care, but in a day or so as the patient's being diagnosed, that's ideal. So Kendall, this is actually her PhD project. She took this on. So what she's been doing is creating a microfluidic device that enables analysis of leukemia cell response to 3 drug combinations. And 3 drugs is really important because that's kind of the standard. So when someone's diagnosed with leukemia right now, the first thing we do is give them this kind of salad of three drugs. So what Kendall did is she created this microfluidic device where she has these 3 ports, and she can perfuse in 3 different drugs, and she sets it up such that there's a concentration gradient that's stable of all 3 drugs. So what she's able to do with those 3 drugs within one patient sample and one device, she could actually get every single combination of these 3 different drugs to get a good sense of the overarching drug response. And what she does here is, so these are actually images, these are fluorescently tagged molecules to give you a sense of what the gradient looks like. She's done CFD, comparing it to experimental data. It looks really good. And then this is what the gradients look like. And what she does is she then takes the data and transforms it into a kind of a geometric simplex such that she could look at things as a function of drug concentration in kind of this more simplified geometric space. So what she's done now, and I'm zooming, she did a whole defense on this. There's basically, I zoomed past like 40 different slides of hers. But what we think she can do is, with her system, she can potentially predict clinical outcomes at diagnosis in pediatric leukemia. That's really important because right now, we still kind of treat pediatric patients who are diagnosed with leukemia, we treat them all the same in the beginning. And then we do some tests of clinical outcome metrics. So there's one very important test called whether or not the patient has MRD, minimal residual disease. That means we give them chemotherapy and let it kind of kill the cells, and then we do some testing to see, hey, do we still detect any small trace of disease? And if we do, that's a bad sign. So MRD positive is a sign that, this patient may actually relapse. This patient's cancer may actually come back. So what Kendall did is we have these bank specimens. She had a good number of patients who are MRD positive and who are MRD negative. And then what she did was she put those different cells in her system. I'm not going to go over this too much, but what she can look at is, so here's her what she sees. She could look at single drug responses. She could look at pairwise comparisons of how cells are killed. And she could also look at 3-way combinations to see if there's any synergy between the drugs and at what doses. So long story short, she looked at a good number of patient samples, and what she's found is there is interesting data that kind of separates readouts between the MRD positive patients and the MRD negative patients, and looking specifically at how, so there's 3 different chemos here. There's one called daunorubicin, vincristine, and prednisolone. Those are the standard therapies that patients with acute lymphoblastic leukemia get in the very beginning, what's called upfront. And what she's found is that she has data that shows that she can start to delineate which cell samples correlate with MRD positivity and MRD negativity, and specifically looking at the response to vincristine, and then looking at synergy and potency. So V plus P means how much synergy exists between vincristine and prednisolone, and she again sees a signal between the MRD positive and MRD negative. So moving forward, we're really excited about enrolling patients prospectively, right? So then we could start to, at diagnosis, get this data. And then we'll see within a few months, a year or so, whether or not the data pans out prospectively. If we can actually identify who is MRD positive or MRD negative in the very beginning, that's a huge game changer, because that means, oh, we'll need to fine tune. Maybe we need to add drugs. Maybe we have to give a different dosing. And Kendall's system can also be used to look at experimental drugs as well. So she's very excited about it. And one could envision that this would be in the clinical laboratory in the hospital, where as the patient's being diagnosed, they could get this, the hematologist, the oncologist, and the hematopathologist can get this data. Okay, so that's the white cell stuff. Now I'm going to jump to some microfluidic studies we've done on red cells. Okay, so COVID-19, we've all heard about it. And this is data from Dr. Ifrig, an MD PhD, and she's a critical care fellow, meaning she's learning to be an ICU doctor in collaboration with Kirby Fibbin, a graduate student in the lab. So what Liz knows is that when you have a patient who is in ICU, and this is from the literature actually, their red cells tend to clump together. We really don't understand why, and we really don't know what the consequences there are and what's clinically relevant. But this is a known phenomenon. So she took some blood, and this is back in the days of severe COVID, so you guys might remember the variants. This is during the first wave, where there's a lot of patients that were in the ICU. And what she saw, this is just a thing of blood, 2 tubes of blood. She looked at healthy blood compared to COVID blood, and she just looked at it. And she said, wow, after 15 minutes, that COVID blood drops, the red cells aggregate together and they drop like rocks compared to the healthy blood. And she says, wow, what's going on? She looked at it under the microscope, and again, she sees these clumps, these aggregates of red cells in COVID-19 blood samples. What's going on there? So what she did was take these blood samples and look at them in the microfluidic. And what she saw is these clumps. And these clumps, when they're perfused under flow conditions that are physiologic, she sees these kind of clusters, these aggregates that form that don't really exist when she's looking at healthy blood. So these clumps that are kind of perfused through our microfluidic system. So this is likely what's going on in vivo. Okay, so what are the consequences of that? So she developed a custom image algorithm that detects, tracks these red cell aggregates, and she then even compared it to other critically ill patients that didn't have COVID, so patients with sepsis that was non-COVID. And she actually sees that there's a lot of differences in these kind of clusters. And most of these clusters occur in COVID compared to healthy, compared to non-COVID. And the sizes are bigger, and also the velocity tends to be different as well. So that's kind of interesting. So she sees this distinct separation between COVID, non-COVID sepsis, and healthy. So what's driving this red cell aggregation? So the hypothesis that Liz had was that there's a protein called fibrinogen. And fibrinogen is in the blood. It's a clotting protein. It goes up when people are really sick, when people have a lot of inflammation. So that was an obvious hypothesis. And what Liz did, she said, well, let me just take some normal red blood cells, and let me just take some normal fibrinogen and just crank it up to what is seen in COVID-19. And what she's definitely sees is this, even with normal red cells, when she cranks up the fibrinogen, she starts to see these giant clusters in our microfluidics. So, okay, well, what does that mean, right? So when you get these aggregates, what does that actually mean physiologically, right? Why is that bad? So what she then did was take some of our microfluidics, and she grew endothelial cells in them, and I'll talk about that a little bit. And she realized that when she perfuses these red cells that are aggregated, there is a protective layer called the glycocalyx of the endothelial cells. It's just this kind of sugar layer that is protective. That is removed when she perfuses red cells that are in aggregate form compared to red cells that are not in aggregate form, healthy red cells. So that removal of the glycocalyx, that's bad. So that is known to correlate with really bad disease in terms of ICU, needing respiratory support like mechanical ventilation, the clotting that you guys might have remembered hearing about in COVID-19, it's all related to that. So that's what Liz discovered. So in many ways, what she's found is that COVID-19 is a red cell disease. So I'm a hematologist, so I'm just showing my bias there. Okay, so I'm going to switch gears a little bit more, and I'm going to go back in time. This was, again, another experiment that I did as a grad student many years ago. I took just drew a tube of blood, normal blood, and then I went to one of my patients I saw in my clinic who had sickle cell disease, did the same thing. Drew a tube of blood, perfused it in the same type of microfluidic, and I was like, ooh, there's some differences here, right? So that really meant to us that there is an opportunity to study sickle cell disease. So what's going on in sickle cell disease is there's a genetic mutation in which, of hemoglobin, in which the red cells become much stiffer. They become stickier too. So it's a plumbing problem. It is harder to perfuse red cells from sickle cell patients at the same conditions in the same type of device. Okay, fun fact number one. Analyses of microfluidic data can be a huge pain in the ass. If any of you have ever done it, I mean, I discovered that, here I am making these chips. I'm going in the fab room. I'm like, it's pretty high-tech stuff. But when I'm analyzing, I was like, I'm taking out a notebook with a pencil, and I'm counting each cell. And I remember talking to my other colleagues in the microfluidic space. They're like, oh, yeah, we do that too. Yeah, what type of pencil do you use? Yeah. So what we did, again, fast forward. We said, okay, let's change this. So we developed our own software just last year. And this was Meredith Fay's PhD project. She's now a postdoc at Duke. So she developed what she calls iClots, interactive cellular labeled observation and tracking software. You can tell it's one of those acronyms that was thought of before the actual words. But the goal is to make for simpler and automated analysis of microfluidic data. So for example, what I kind of just showed you, cells perfusing in these little channels, you just need to upload the video, and the software automatically spits out these metrics. It can count the cells, it can tell you how fast they're going. And what Meredith did was even create some other functionality as well. You could look at how well cells are sticking to a surface. You could look at accumulation of stuff in microfluidics. You could look at the velocity profile, clustering, again, similar to what I showed you, and then really easy video editing. So this is all meant to be very easy to use. It's completely open source, so you could download it here. And we have our methods as standalone scripts on GitHub, and source code, opportunities to contribute, again, open source for anyone who wants to play with it. So we're really hoping that this could help the field. I know I would have loved it as a grad student. Okay, so I alluded to this a few minutes ago, but there is a technique that our lab has become really, really good at, and it's not me anymore, it's the guys in the lab, where we can grow blood vessel cells, endothelial cells in microfluidics. And that allows the system to be much more physiologic, because when you take a typical PDMS structure, well, it's not quite like what's going on in the body. But now when we grow blood vessels in there, then the blood is starting to see an environment that becomes much more similar to what's going on in the body. So we can grow endothelials cells in these systems. This is what Liz used for her glycocalyx experiment I described a little bit about. And we can get down to the size scale of the microvasculature, the small blood vessels. And that allows us to really look at all these different interactions between the blood and endothelial cells. It's important for sickle cell disease. Here's an set of images using confocal microscopy to give you a sense that these endothelial cells are growing and spanning the entire 3D surface of these microfluidics. So sickle cell disease, I told you about, is not only a disease in which the cells are much stiffer, but they're also stickier, and we're able to recapitulate that. In fact, you see some interesting differences between these two conditions where we're perfusing whole blood from a sickle cell patient, and then we're also perfusing whole blood from a sickle cell patient, but this patient happens to be on a medication that attenuates many of the symptoms that those patients encounter, a drug called hydroxyurea, and that kind of recapitulates that. So since these are all done via video microscopy, we now can create and develop a lot of different types of data, analyses, especially now that we have iClots. So moving forward, ongoing questions, and a lot of this is being done by Dr. Christina Caruso in the lab, really looking at issues of how cell interactions play in the pathophysiology of sickle cell vasculopathy. Vasculopathy is a disease of the endothelial cells. How does that really happen? How do these cell-cell interactions correlate with clinical outcomes? We're very interested in that. And then even using this type of system to discover how drugs work. The FDA has now approved a good number of drugs. In fact, just the last week, they approved 2 genetic therapies, gene therapies. There's a need to really create systems that can serve as biomarkers for whether or not those drugs are working. So we're very excited about that. Okay. So now I'm going to switch gears again. I talked about white cells, talked about red cells. I'm going to talk about clotting and platelets. So there is a need for new type of bleeding tests that incorporate all aspects of hemostasis. Hemostasis is the process of clotting. So right now, if any of you, hopefully none of you, ever encounter a really bad bleed and the physicians try and diagnose you, when they draw blood, they can only look at all the different parts of blood clotting independently. But in reality, it's this very complex orchestrated set of events where all this stuff, they're not happening in isolation, they're all interacting with each other. So there's a need for new types of tests where you're really looking at all the combined aspects of blood clotting. Now, there's some older school ways to do it, right? If you're really trying to figure out how well a person is clotting, well, I suppose what we could do is cut you, right? And you would argue, well, that's not really, it seems like there's a lot of variability. I'm like, that's true. So we can cut you multiple times, right? And therefore give you more statistics. We don't want to do that. It's now known to be pretty medieval and actually doesn't work all that well because of different users, right? Different users with a knife. If I cut Dr. Gottfried, maybe you cut him as slightly different force. Maybe you like him less, so you'll use more force, right? So sorry, David. So how can we take what we know about microfluidics and take what we know about blood clotting and create a kind of a model like this, right? Where can we create an in vitro model of a blood vessel that's injured and then just kind of stare at it and look at how all the different components interact. So Dr. Elisa Hardy, who got her PhD here in electrical engineering and is a fixture in our lab and in the IEN, those of you that use the clean room here see the good Dr. Hardy quite a bit. She interacts with a lot of IEN staff. And her partner in crime, Yumiko, created this really neat device where we use a process called pneumatic valves in which we use one layer of PDMS to function as a valve, as kind of a trap door. So the way it works is this is a cross-sectional view. So if you see my pointer, the blood is kind of going into the screen, and then we have a pneumatic valve that we can pull on, and then it pulls and it opens. So it's kind of a bleed. Blood leaks out that way. So if you look at the top view, what you have is blood goes from left to right, and then when we activate the pneumatic valve, the blood leaks, and it's under a video microscopy. We watch the whole thing. So here's an example of how this works. So what you'll see is blood will come from left to right, and a whole bunch of gray stuff will come flushing through, and those are red blood cells. Blood is mostly red blood cells, but you'll start to see platelets in red. You'll start to see fibrinogen, and I told you about fibrinogen with Dr. Iverick's stuff. You'll see it in green, and these are endothelial cells already stained in blue. Okay, here's the vascular channel. Here's what we call the bleeding channel. So everything's kind of spewing out. You start to see these platelets collect and aggregate at the site of the injury, which is what you expect. And then you start to see the fibrin start to coalesce there. And what fibrin does is it's actually the polymeric form of fibrinogen. It acts as a glue that kind of glues all the platelets together, glues the clot together. So what we're able to show here is that we can create a model of bleeding and the clotting that causes cessation of bleeding. So you see everything kind of stop here. And it stops in about a few minutes or so, which is pretty physiologically relevant. So now we can start to look at patient samples, right? If I look at a healthy control, and what you see here is the response of, so you see the injury here at the endothelial cells, you see the nice platelet response, and then you see the fibrinogen response or fibrin response. In contrast, this is whole blood from a hemophilia patient. And patients with hemophilia, they lack the gene that allows fibrin to be formed, or at least formed well. So what you see is kind of what you expect here. You see a nice robust platelet response, but you see zero fibrin. And then you can start to time things and kind of basically do a bleeding time, kind of like what we would do if we cut you. So now we don't have to, right? We could do it in a microfluidic. And what we show here is, sure enough, that when you have healthy people, this clotting occurs in a certain amount of time. And then when you have patients with a bleeding problem, it's much higher. So we're very excited about that. And now we're using this model as a drug discovery platform. So for example, we can look at the healthy control. You see the red platelet response. You see the fibrinogen response. And then we can use, for example, an antibody that makes the normal blood into more of a disease state. We have an antibody to what's called factor VIII. Factor VIII is actually what's missing in hemophilia. Without factor VIII, you can't make as much fibrin. And again, you see that. So now we could start to use some candidate drugs to kind of say, hey, can we use some medications to push it from this state closer to that state, rescue it? And we see that. So this is exciting, and we now have several contracts with several pharmaceutical companies to look at their drug of action. They like our system because it saves them money. They don't have to spend money on big animal studies, and sometimes they even use our device as a way to inform them about their animal and human studies. So fun fact number 2, for anyone who is in the microfluidic game, that biotech and pharma companies can actually be a good source of funding for microfluidic studies. Okay, so changing gears again. So I told you that microfluidics that are just made of PDMS, they're not that physiologic. They don't mimic the in vivo situation that well. We grow endothelial cells in them to make it a little bit more physiologic. Now, what we're really trying to do is we say, okay, let's get rid of the PDMS altogether. I mean, PDMS is not physiologic at all. Can we take something that's more tissue-like to be the substrate for our microfluidics? So this is done by Yang Xu in our lab, a brilliant and assistant professor who's a great bioengineer in our lab. And what he is starting to develop is this assay that is much more physiologic. So what does this buy us? What does it give us? Well, it allows us to do many things. First of all, the endothelial cells are really happy in here. They can be grown for several months for a device. And we could also start to look at how leaky the endothelial cells are. And then we could start to look at how these phenomenon I've been describing to you before, like in sickle cell disease and clotting, how they resolve. Now that we can look at things over days, weeks, sometimes months, that's a different time scale than we're typically used to with an assay. We could start to look at phenomenon that occur in the body that occur at that time scale. So Yang, first of all, characterized the system, really showing that endothelial cells are super happy in this device that is not PDMS, but is a combination of collagen agarose. It's a hydrogel. and they express these what are called basement membrane proteins. So they're basically setting up shop. They're saying, hey, I'm in the human body here. I'm going to do what I'm supposed to be doing, and really laying down a matrix for them to grow on. And what Yang is now able to do is monitor, for example, a blood clot. So what he has here, just perfuses whole blood here. You see all this gray, those are our red cells. And if we flip over to a fluorescence channel and we have antibodies to things like fibrinogen, platelets, and neutrophils, well, actually, we don't see much, right? Because this is a healthy condition. Now, on the contrast, if we make the endothelial cells, if we piss them off, if we make them sick, we expose them to a chemical that occurs under inflammation, and in this case, it's a chemical called tumor necrosis factor. Now we can see these clots form. We can see these clots form, and they stick around. So what you see here is both fibrinogen in blue, the platelets in green, and then if you wait long enough, you even see some neutrophils kind of sticking around in red. That is what we expect. A blood clot forms. Now, to take advantage of this system, Yang could now look at how we make the clot, and then he can keep looking at it, and he can start to perfuse normal plasma from blood, and then start to look at how the clot goes away. Because that's what happens in real life, right? If you have an injury, you got a blood clot, it doesn't stay there forever, it goes away. But there's not great ways to study how the clot goes away. Yang is starting to address that issue, where he has these timelined experiments. These are timelines that show his experiments that last for numerous days, and he can kind of keep track of where all the different events are. The platelets, when they stick together in a clot, the first day, and then when they start to go away. He can look at fibrin, and when does the fibrin start going away? And it looks at neutrophils that stick around. Do they become persistent? So this whole timeline is really stuff that people haven't been able to do because of insufficient techniques and technologies. Yang's really starting to scratch the surface of that. We could, again, use this system to look at how drugs work. So those of you might have heard of TPA. It's a clot buster drug. So if any of you unfortunately know anyone that had a stroke, that's the drug that physicians try and give within a certain window. And what Yang used was, okay, well, let's look at a clot here and then perfuse TPA. And we see a lot of things that we expected. The platelets completely go away, the fibrin's completely degraded. But what's interesting is, okay, the neutrophils kind of stick around. That's neat. But what's also neat is that the endothelial cells kind of also get better. So that's an observation that has been made before. So using a standard drug, using a standard kind of clot formation process, Yang's able to make some observations that are pretty novel. So we're very excited about that. Okay, fun fact number 3. When a blood clot forms, platelets actually exert muscle-like contractile forces, and it dramatically shrinks the clot. Clot, missing a T there. So can we use microsystems, microfab devices to study the underlying biophysics of platelet-mediated clot contraction, right? So if I take a tube, just a random tube, regular tube, and I put platelets in it, I put fibrinogen, again, it goes back to that chemical I told you about, and then this other enzyme called thrombin that both activates the platelets and also makes the fibrinogen into a polymer. What you see is a blood clot here. In this case, this is a square tube, and you see it shrink over 25 minutes. It's shrinking, 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 because what's going on is the platelets are activated, and they grab onto the fibrin, and they pull it. So the blood clot actually gets down to about 10% of its volume. This is what's going on in the body. It's kind of neat. We still don't really know exactly how things work, but if you look at it under a microscope, what you see here is a platelet in red, and it's kind of pulling against these fibrin strands. So you go from macro to micro. So we had this question. We said, oh, wow, if this cube kind of sticks in this same shape, does it mean that like all the platelets are pulling the same amount in every direction? So what we did was we said, okay, let's take a mold, random mold. In this case, we had a mold of a Lego man, and we made a little clot. And we said, okay, let's activate the platelets and see what happens to the Lego man. Does the shape of the Lego man stay the same? And it turns out it does, right? So, and luckily there was a little bubble on the head of the Lego man to keep him upright. Otherwise, it would have fallen over. So it's pretty fortuitous there. But what this means to us is that clot contraction is happening in an isometric manner. All the platelets are pulling the same amount of force in every direction. So that's kind of neat. Okay. But what we really wanted to do is look at things at the single cell level. We wanted to actually measure how strong a single platelet is. So this was work done by Dr. David Myers, who again was a fixture in the IEN. He's now runs his own lab over at Emory, but still collaborates with IEN folks quite a bit. So during his postdoc in our lab, he developed a technology called platelet contraction cytometry. And what he did that was brilliant was he took a hydrogel of known mechanical properties, and he put these little dots of fibrinogen on, and then when a platelet would land, it would extend one of its little arms, called filopodia, and attach the other one, and then it would apply this contractile force, and since we would track how much these little dots would move closer together, and since we know the material properties of the underlying hydrogel, we can use Hooke's law, right? You remember Hooke's law from high school, right? F equals kx. So we know the k, and we're measuring the x in terms of displacement of these dots to give us F, the force of contraction. So what we discovered that's pretty neat is that platelet contraction occurs nearly instantaneously upon contact. So when a little cell grabs onto both dots, it starts pulling, and it pulls like crazy. A little platelet can contract with as much force as a muscle cell. And if you think about the size scale difference of a platelet is about 1/100th the volume of a muscle cell. So that's pretty fascinating. I mean, it really means that nature has designed these little tiny cells to be contractile machines. And what we're also really fascinated by is that when you change the mechanical properties of the underlying substrate, they pull at different forces. The stiffer the underlying hydrogel is, the more they actually contract. So that's pretty fascinating. And other cells do that too. So it's neat to see that there's some conservation of that. Here's an image of what an experiment looks like. You see these little red platelets that are falling on this matrix of these little dots. You see this platelet here landing between these dots and pulling. And then we actually zoomed in on here. So we took away the platelet. We're just looking at these two dots moving close together. It's kind of hard to see with the naked eye, but luckily David has a nice code that does it all automatically, and it gives him a sense of all these. He can create these histograms where we vary the amount of this chemical. It's called thrombin. That's what activates the platelets, and we vary the stiffness of the gel. And he can do this, many, many times and get these nice histograms. So our question though was, can we use this system now as a potential clinical biomarker for diseases, specifically for platelet diseases? So when David got these two sets of data, we were really intrigued. So we get this histogram of a healthy control where you have all these platelets with all these different forces, and it's never normally distributed. So it's pretty fascinating how there's all these subpopulations. Then we take a patient who's known to have wimpy platelets. So there's a set of patients called Wiskott-Aldrich patients, and they have a genetic mutation in the platelet contractile machinery. So we kind of see this, that's expected, right? You see the peak here, it suddenly moves to a much lower force. So that's like almost our positive control. Then we had a set of patients that were bleeding, and they weren't diagnosed with any real disease yet. We said, ooh, look at that. They are kind of in the middle. And we did a lot of characterization to try and really figure out how contraction force correlates with other markers of platelet biology. Never really found anything, frankly. But what we were intrigued by with those patients in the middle, they all ended up being diagnosed with a disease called immune thrombocytopenia. Okay, so what is that? So that is a disease in which your immune system temporarily attacks its own platelets. So patients have a very low platelet count. They're at risk for bleeding. About 10% of them have major bleeds, and then 0.5% have really life-threatening bleeds because it could occur in the head. However, most patients don't end up that way. Most patients are fine. You don't actually have to do anything. They just get better on their own. But we as physicians are always stuck because there actually is no biomarker that reliably predicts who's going to be in the good 90%, who's going to be in the bad 90%. Therefore, we don't really know who needs treatment. And some of these patients do, unfortunately, become chronic, like they don't get better. So we need more clinical tests to help us figure out who in ITP needs. We need more biomarkers for ITP. So what we did was we got a bunch of ITP patients, and we all put their platelets under the cytometer, the platelet contraction cytometer. So here's a couple of patients, a couple of healthy controls. That's what their histogram kind of looks like. Here is a couple of patients with ITP. They have very, very low platelet counts, but they don't actually have any bleeding. There's no signs of bleeding at all. Remember, I told you most of these people are fine. We have then two patients, again, diagnosed with ITP, but these patients do have bleeding. They have bleeding, and then what we found is, okay, these patients with ITP, so if you really look at these 4 histograms on the right, the major difference is that patients who are not bleeding have this kind of hypercontractile, very high force subpopulation that the patients with bleeding do not. So I said, that's interesting. So this ended up being Mo Oshinowo's PhD project, and he just is graduating, and he will walk this weekend. And here's 40 patients from his cohort, all diagnosed with ITP. And what you see are patients who are bleeding, you're able to separate them out. Patients with bleeding have these much lower forces, these wimpier platelets than patients diagnosed with ITP that don't have bleeding. So that was kind of fascinating. So then we kind of looked at, okay, let's look at things that clinicians normally use right now. So we use platelet count because some of these patients develop disease, but it's really just their platelet count's low, nothing else is going on. And then we also looked at the bleeding score. So there's a clinical score, 0, 1, 2, 3, 4. 0 is no bleeding. 4 is like, oh man, he had to go to the ICU because he or she had a really bad bleed. And then we looked at force. So we mapped them all on one graph. And what we found out is that all the patients that had high bleeding scores, 3 to 4, all had fairly low platelet forces and also low platelet counts. So we said, that's pretty interesting. So now let's do what's called an ROC curve. And I think you guys in engineering, it's done quite a bit in electrical engineering, where we look at the sensitivity versus specificity. So if you look at a horrible test, like no better than a coin toss, it would basically have this kind of diagonal line. An ideal test makes like a right angle, where it's both very sensitive and also very specific. Because in general, when you improve one, sensitivity or specificity, there's a trade-off, you lose the other. So the ideal test, you don't lose anything. So what we found is that when you just use platelet counts, which is the only thing that physicians use right now, it's not all that great. And then they also use something called platelet volume. There's a thought that big platelets might actually be more active. Again, not so great. But platelet force, however, seems to do okay. So we actually see a sensitivity of about 100%, specificity 84.2% to detect bleeding using only contractile force. Now, when we couple that with another marker like platelet count, we could increase our specificity to 95%. So what Mo is doing now, even though he's graduated, he's still got a few more months, is trying to figure out what the mechanisms are. We could go over that in a bit, but we actually think it's related to antibodies and where on the platelet the antibodies, remember I told you it's an autoimmune disease, where the antibodies actually stick to the platelets. Okay, got a few more minutes. I'm going to jump into some of our diagnostic tests that we've actually kind of started to bring into the real world. Erica Tyberski, who is part of our ACME POCT that David told you about. She's a regulatory expert, but she's also a brilliant scientist. Her capstone project, when she was an undergraduate here, she designed an anemia test. It's a chemical solution in which she can tell whether or not someone is anemic based on the color of change that the anemia test undergoes. So it's very easy. Drop of blood goes into her little vial, and then based on how severe the anemia is, so very severe compared to healthy, you get a different color. And pretty decent sensitivity and specificity to detect mild versus severe anemia. And that has now been licensed to our startup company, Sanguina. It was cleared for clinical use in 2017, and then just a couple months ago, FDA cleared for home prescription use in September 2023. So Erica is very excited about that. She did a great job doing that, and she's the CEO of our company. Now, we have another technology developed by Rob Menino. He was both an undergrad and a PhD student and a postdoc in our lab. What he did, he created something completely non-invasive. So in medicine, we know that patients with anemia tend to have pale body parts. So the fingernails are kind of pale. What's called the conjunctiva right under the eyelid is kind of pale. The creases in your palms can be pale. So Rob actually developed an app where he determined that the fingernails were pretty much the most convenient place to do it yourself. So a fingernail selfie can help estimate whether or not you have anemia. We don't need any blood at all. It's completely non-invasive. Long story short, it kind of works. Sensitivity is not bad, specificity is not bad. Again, there's no need for any blood draw. This has now been folded into our company as well, and we're trying to figure out the best way for both products to synergize. Now, NemoCheck is what we call the app, has been released into the real world. You can download it yourself. You can go to Sanguina if you want. It's on a freemium basis, so you get a handful of free tests, and then Sanguina will charge you. I actually can't do it myself anymore because I'll be charged. So it's been used about 102 million times in the US. And this is where we said, okay, let's be scientists here. Let's really look at this data. So if you look at where we are, 1.2 million tests in the US, we can get a map of anemia. And if you look at where everything is, it just looks like a population map, right? The most dense areas, New York City, LA, Chicago, that's where we see the most uses. That's what we expect. But if we control for population density per capita, that becomes interesting, right? We're able to see in terms of places, where are places disproportionately more anemic than other places. So then it becomes interesting. So we actually found that Florida is our most anemic state. It's always Florida, right? Why is it Florida? Weird stuff always happens in Florida. And then we even asked the question, is it more rural? Is it more urban? Like, not really, right? If you look at where that is. So we're still trying to figure this out. But we can make some interesting observations. When you got something that's been used 1.2 million times, there's a lot of interesting data here. So one question I had for the group was, okay, we got data between Android and iOS, right? So what's the difference there? We're like, whoa, look at this. So again, many, many uses. People with iOS versus Android, they seem to have different distributions. Now, you can make the argument, well, maybe Androids just suck, right? I'm an Apple user. But these patients, a proportion of these patients also submitted lab tests, right? These have nothing to do with us. And the distribution is pretty similar. So that's kind of fascinating. So in general, Android users have lower hemoglobin levels than iOS users. Now, why might that be? We have some hypotheses. So we actually think it has to do with socioeconomic status. So people who are lower socioeconomic status tend to have Androids. And people with lower socioeconomic status have more chronic illnesses. Chronic illnesses often lead to anemia. So we actually think this device is great for public health, for surveillance of disease. So we're pretty happy about that. Okay, last couple minutes, I'm going to talk to you about one last project. And this, again, I could talk for 2 hours. But it relates to cell manufacturing. And I wanted to bring this up because of what I alluded to a few minutes ago. There are now cell therapies, gene therapies that are coming around the bend. The problem is they're really expensive. So there's these things, for example, called CAR T cells. I'm not going to belabor this, but point is, it's a complicated process where you take a patient's own cells and you muck with them, you engineer them, and then you put them back into the patient so they can kill bad cells. In the case of what are called CAR T cells, patient T cells are modified, engineered, put back into the body to kill cancer. And there's a huge engineering manufacturing process. In fact, our own center here at Georgia Tech, we have a center called CMAT. It's dedicated to trying to make this whole process a little bit more efficient. This is Reggie Tran's project. He started as a grad student. He's now, he did his postdoc on this project, and now he's a faculty member still working on this project. Point is, cell manufacturing requires a lot of virus, and this is expensive and supply limited, and this process of getting this engineered virus to make the cell that you want, what's called viral transduction, is a very inefficient process. So what Reggie did is for his PhD, he created a set of microfluidic, and I call micro because some of these are actually, they're not so micro, they're pretty big, But they're fluidic platforms that overcome some of the efficiencies of these existing systems. So he calls them Proxim, and he's got a whole suite of these different devices, anywhere from like small scale that can fit on like a 6-well plate for those of you guys who do any biology work, to bigger devices that can really span clinical scale. Like he can put in like 10 to 8 number of cells in these things. And what he's shown is that his devices are much more efficient than the standard platforms that you see in red. I'm going to hurry just because I know I'm running out of time. So how does the whole thing work? So in a typical cell manufacturing process, people use these cell culture bags, and they're really good at nutrient delivery, and they're really great at allowing for space for growth. But there's dead volume. The dead volume is part of what makes these so efficient. So the fluid heights actually waste virus and increases the time needed for transduction. Now, what Proxim does is actually minimizes the height to maximize viral transduction. So it's actually it's one of these pretty simple but elegant things that Reggie came up to. That's how Proxim works, right? You got these viruses floating around, just brings them closer together. And what he's shown, and this was all part of his PhD, is that you really can get much higher transfection. So GFP is just like a dummy gene that makes the cell glow green. You get a lot more cells that take up the virus in Proxim than a typical type of platform. In this case, he's comparing it to 96 wells. So what he's done now and what he's trying to do is create not only the hardware part, but also the analytics part. And when you start to get down to the level where you maximize efficiency, that's where things get interesting, because then he can kind of analyze different protocols. And this is really important because the FDA just came out with this a few days ago. In the context of these new types of cell therapies, there's a worry. There's a worry that some of these might cause cancer, and I'll tell you why. So this is very convoluted, but this is where Reggie kind of sees where everything fits together. So when you have a gene therapy protocol, there is something called VCN. VCN stands for vector copy number, and it's the number of viruses that go in per cell on average. You need a high VCN to show that your viruses are actually being taken by the cells. However, the problem is when you get too high, too much virus that's integrating into the genome, that may actually cause cancer. So the FDA said, okay, we don't like anything that's greater than 5 average VCN. Now, how Reggie, however, because he has his hardware and because he's been paying attention to these cells, he knows that the cells actually don't behave in an average manner. They behave in a Poisson distribution. And because he's able to take these cells and maximize their efficiency, he now can take any protocol and he can say, hey, you're here, you probably want to maintain your same average VCN, but you don't want these high number of VCNs, right? So he can follow the Poisson distribution. And the bottom line here is that with his analytical platform, and again, I'm skipping over 20 to 30 of his slides, but what he can do is he can take any protocol and he can learn about that protocol. He can figure out the transduction dynamics when he's combining Proxim with his software. He can tell you how to optimize your dose, and then he can even tell you how you might want to tweak your protocol to make it the safest, but also the most efficacious, and also the cheapest, right? So Reggie's technology we're very excited about and has quite a bit of legs. I skipped over so much stuff, but this is just to give you a sense of what you can do. I'm going to give you my acknowledgments. There's a bunch of other projects I didn't go over, but those names highlighted in red are the projects I did highlight. So I'll stop there, David. Probably have time for a couple questions. Thank you.
Time for maybe 1 or 2 questions.
Thank you so much, Professor Lam. So I was curious, what was the problem with the PDMS version of the endothelial growth device compared to the hydrogel version? Like, why was it not suited for elongated experiments like the other ones?
Yeah, so we have a few hypotheses, but the bottom line is when you grow endothelial cells in a PDMS device, they grow, they can maybe replicate 1 or 2 times, and then they stop. They just die. We have a few hypotheses. One is that the substrate in and of itself is not that great, right? There's, and others have characterized this, right? There's leaching of biomolecules that can go into PDMS, and PDMS also is physically very different than what a tissue is, right? Even if you look at, you characterize it in terms of an elastic modulus, it's well beyond several thousand kilopascals, whereas in the tissue, you're really talking about 1,000 pascals. So Young's hydrogel is not only more relevant biologically because it has collagen, but it's also softer. So that is one capability. Now, your question is a good one. We're still trying to figure out the question, what makes this hydrogel so effective in making endothelial cells happy? Question back there?
Of course.
Thank you for your talk. So I had a question about what you mentioned about clotting and seeing more clotting in the COVID patients. So one of the things that I was curious to know more about is, were there any other factors that the COVID patients had in common, like for example, medications that we're taking or like types of treatments that they were receiving? Were there any other confounding factors that may have contributed to that?
Yes, absolutely. I mean, a bunch of stuff is probably going on with them, and that's why I think we're just scratching the surface. I mean, you'd have to really do a true kind of controlled experiment, but practically that's going to be pretty hard. So would other things have caused that? Maybe, maybe not. I think what really lends some credibility to Liz's data, though, is that she sees quite a big difference between COVID versus other diseases with sepsis, of which those patients probably got roughly the same thing. They were all mechanically ventilated. They probably all got support for their kidneys and other organs and so forth. So there's definitely something going on with the red cell stuff in severe COVID-19. The question is really, does that answer everything? Probably not.
All right, I think in lieu of the hour, we're going to end up here. Let's thank Wilbur one more time.
Thank you all.